And we are now recording, and thank you very much, Dave, for agreeing to give a Build a Cell seminar, and it's all yours. Very good, thank you for the invitation. And good morning to the Build a Cell community. I'm very happy to be here. This is going to be a little bit different. I'm going to start with about a 5-minute demonstration of how you can put together a cell from its isolated components. And it just takes literally a few hours to do this. It's something we've experimented with. And I think that you'll enjoy seeing how easy it is to do this. And at the end, I'm going to invite all of you to try this out yourself, because all you need is some of this. This is 𝗕𝗂𝗶𝗹𝗶𝗶, but any lipid that self-assembles into membranes will work. I'm going to show you the structure of this when my slides begin. This is 20 milligrams per ml in chloroform. What I've done is to take half a milliliter of that and put it into a test tube. Now, you're not going to be able to see it, but there's a dried film of phospholipid down toward the bottom of the test tube, and it's 10 milligrams of that phospholipid, 𝗕𝗂𝗶𝗶𝗶𝗶. Now what I'm going to do is to add 5 milliliters of pure water. Uh-oh. Excuse me, my water disappeared. I'll be right back. That happens more often than I care to admit when I set up an experiment and I think I have everything ready, all lined up, and then I get going and like, oops, I'm missing this one thing, or something spilled, or I forgot about something. I know. Hi, you're back. Little quick trip to the sink in the next room over. So there's my water, and here's the lipid. As I say, you can make liposomes just like Alec Bangham did in 1965. And these are called hand-shaken liposomes because you literally shake them by hand. And what you're going to see is the lipid becoming dispersed as a cloudy, milky suspension. Can you see that beginning to?
Is that my coffee from before?
Say again? I think someone didn't know he wasn't muted. I just muted him. Oh, okay. So if I keep shaking this, it's going to become dispersed. And I'm going to end up with 2 milligrams per milliliter of lipid in this cloudy suspension. But then what I've got to do is to sonicate it. And as I say, I'm going to show you images before and after sonication. So here's that same preparation after sonication, and you can see that now I can see my finger. It's become translucent, so you can see my finger right through. And the reason is that with about half an hour of sonication, those large globules of what we call multilamellar hand-shaken liposomes have become dispersed into small unilamellar vesicles, and we abbreviate that SUV. They're only about 200 to 300 nanometers in diameter, and they are single membranes on the outside, whereas that cloudy suspension I showed you was a multilamellar onion-shaped globules of lipid. So we're going to use this as a start. And what I'm going to do is to show you how you can encapsulate molecules as big as DNA inside the liposomes. And the reason I'm showing you this is that you can also do this with the cytosolic components of a living cell. So we have, in fact, popped cells open, bacterial cells. We have captured them in lipid vesicles, and you're going to watch and see what happens when we do that. Okay, before I got started here, I weighed out 2 milligrams of this. And this is a DNA isolated from salmon sperm And it's really inexpensive. You can get all you want for just a few dollars. So I'll just pull out some of it for you. If you've never seen DNA, it's quite extraordinary that we know that it's long, stringy stuff, and there it is. This is salmon sperm, and it contains the genomic message required to make a new salmon. This, of course, is the male half of the genome. And maybe if we stuck some of that into a unfertilized salmon egg, something might happen and we could build some salmon cells. Just kidding, of course. Let's try to put that in now. So I've weighed out 2 milligrams. Here it is. You can probably see these 2 little spots there. See those? So they're going to go into our sonicated vesicles. There it is. It takes a little while to dissolve, so I'm not going to wait around to let it all dissolve because these are very long stringy molecules, and this is distilled water, and you can sort of see it beginning to swell in there. So about an hour from now, I would have all of that salmon sperm DNA mixed in with the lipid vesicles. What I'm going to do now is to take a small amount of that. I'm not going to wait. Some of it is dissolved. I'm going to take 20 microliters and put this onto a slide. Here's a slide, just ordinary glass slide. Put the 20 microliters in the center. There it is. So that's now a mixture of the salmon sperm DNA and the sonicated lipid. And I'm just going to let that dry out. It'll take about an hour to dry out. When we do this with bacterial components, we do this under vacuum for overnight at freezing temperatures, basically almost freeze dry it. And under those conditions, something interesting happens. And I'm going to show you that as we begin the slides. Okay, so with that demonstration in mind, I'm going to share my screen and click on there we are. Do you see my slide sharing?
Yep, we see it.
So let's click over to play. Okay, that's a blank screen on purpose. The title of my talk is Building a Cell DIY. And DIY, of course, stands for do it yourself. This does not require a lot of fancy equipment. And anyone listening to what I'm telling you about could repeat this very easily. I'm going to take a lesson from Dr. Frankenstein. And this is a metaphor for what we're all trying to do. 2 years ago, it was the 200th anniversary of the publication of Mary Shelley's book called Frankenstein, a Modern Prometheus. It's published in 1818 in England. And I was giving a talk over at Stanford, an invited talk, and they said that we insist that you include something from the Frankenstein myth as part of your talk. And that's perfectly appropriate for building a cell, except that Dr. Frankenstein wanted to build a person. So we're going to take a lesson from that. Again, my name is David Deamer. I'm in biomolecular engineering at UC Santa Cruz. 2 other people were involved in this. Rachel Lewis did the unpublished work that you're going to hear about today. She was a student of Lynn Rothschild over at NASA Ames Research Center, just across the hill from where I'm at UC Santa Cruz. And this is today. So how does the story of Frankenstein fit into the narrative of building cells? So I'm going to take you on a little history, including a bit of personal history, in fact. I'm going to tell you about 2 young people in love. One of them was Mary Godwin, and the other was Percy Bysshe Shelley. Percy Shelley was a young poet who already was very well known for his poetry that he was writing in his early 20s. He and Mary would have fit in beautifully to UC Santa Cruz because they were very different from the usual sort of English society. They were young, they were exploring, they were innovative, they were both interested in writing, and they fell in love. And there's Mary Shelley in 1840, so she was in her middle years at this time. But when she was 19, she traveled with her new husband, Percy Shelley, the poet. And on that trip, I'm going to tell you how she wrote Frankenstein. Her birth name was Mary Godwin, another well-known English family, and she sort of broke away from her family to travel. And in her mid-teenage, she was probably about 16 years old when all of this happened. So she and Percy Shelley decided to have a tour of Europe. And on the way home, they traveled up the Rhine River. And on the way, they passed a little community called Germersheim. She might have heard of the Frankenstein Castle, which is just a few miles away from where they were. Frankenstein, by the way, simply means stone of the Franks. The Franks were an early tribal society in Germany, and they also gave us the word Frankfurt and, of course, Frankfurters that we've probably all enjoyed once in a while. So here is what is left of the Frankenstein Castle. It's up in the hill near Berg Frankenstein, and that's all that is left after 800 years is just a few of the walls standing up, looking down from a hill on the rest of the nearby German countryside. So in the summer of 1817, Mary and Percy had gotten married. She was 19 at this time. They were visiting Lord Byron, another English writer who had a place to live in Geneva, Switzerland. They were there in the so-called year without a summer. And a volcano had exploded in the South Pacific, and its dust clouded the sun to such an extent that it never quite warmed up during the entire summer, including the time they spent in Geneva, Switzerland. So they were bored, raining all the time. To pass the time, they decided to write horror stories. And Mary might have remembered in her subconscious the Frankenstein Castle from her travel up the Rhine. Here is her original manuscript that she wrote responding to the challenge of Lord Byron to write a horror story to pass the time. She was 19 years old. The first sentence reads, and this is the way it starts in the actual book, "It was on a dreary night of November that I beheld my man completed." This was amazing. A 19-year-old girl writing a book like this. It was published a year later, and she was not listed as the author. The reason was that the publisher just didn't think that it was believable that a 19-year-old woman would have written this very adult novel. So it succeeded beyond all belief. Over the next few years, future editions came out, and she was shown as the author. So what was in her book? Well, Dr. Frankenstein had an idea. He thought that if he could take a couple of arms, a couple of legs, a torso with all the legs and all the organs intact, and of course a head, in this case, the famous head of 𝗕𝗓𝗏, yeah, slipped his mind, Borlaug. who played it in the first 1930 movie, Frankenstein. So Victor Frankenstein stitched together these pieces into a monster, a true creature. And he and his assistant Igor decided to use what we still use today to start up hearts that have stopped beating. That is to say, the monster was there waiting to be brought back to life, and they gave him a very large shock. And indeed, Victor, it's alive, said Igor, and there's Frankenstein's creature, just coming alive in chains. He was displayed as a true monster in this movie, but in fact, if you read Mary Shelley's book, he was a rather sympathetic character. And here is Boris Karloff again, playing the monster. And if you could see what he was thinking about the original creature, he just couldn't understand why nobody liked him. And he searched throughout the world, actually ending up in the polar region, hoping that he could find some place to be. Well, this is what we're doing in building cells, except instead of a creature stitched together from body parts, we're trying to put together a living cell, shown here as a large bacterium, and we're trying to spark it to life somehow. And what I'm going to tell you in the rest of my talk is one way to try to do this. Stitch together a living cell from its parts. So Victor Frankenstein was assembled from a parts list: a head, torso, arms, and legs. Can a cell assemble from a parts list? Well, maybe, that's what we're thinking, but what are the parts? A simple cell like a bacterium has hundreds, if not thousands, of ribosomes. It's got a circular genome. It's got a cell membrane. It's filled with metabolic enzymes. It's got an electron transport system with another dozen enzymes. It's got an ATP synthase to make ATP. It has transport proteins embedded in the membrane to let nutrients in, and the list probably goes on and on. It is a complicated system of molecules. And what we're trying to do is to take it apart and put it back together again to some primitive version of the living state. That's what Build a Cell is all about. It's tough. We've gotten part way, but nobody's gotten all the way yet. Here is an approach that we have tried. What we do is prepare liposomes from bacterial lipids. We decided that we might as well use bacterial lipids from the species that we were trying to reassemble, which would be either E. coli or 𝗕𝗂𝗕𝗂𝗏 radiodurans. Now, if you're a microbiologist, you know that the 𝗕𝗂𝗕𝗂𝗏 radiodurans has this extraordinary ability to piece its DNA back together even after a severe dose of ionizing radiation. So we thought that this might be a kind of a bacterium, that if we wanted to bring something back to life, it would be this radioduran species. We add the bacterial cytosolic components. I'm going to show you how we're going to do that. This is simply breaking them open by a specialized technique. And then you gather together all of the cytosolic components from inside the cell. Now, in what I told you, what I'm going to show you, we left behind the membranes, but we didn't have to. In a complete system, there is an easy way to include the membranes in this sort of a stitching together process. And so we would be able to have electron transport enzymes and the ATP synthase and the nutrient transport systems also present. So keep that in mind. What we do, and this is the step that I think is the key, we evaporate the solution under vacuum in the cold, which allows a concentration of the components from the bacteria, and liposomes. So as they get concentrated, the liposomes fuse into a multilamellar film with the components captured between layers. And I'm going to show you direct evidence that that does in fact happen. Finally, when we rehydrate, you add water back to this dried mixture, just as I can add water back to this dried mixture I put on the slide in this little demonstration. What happens is that the multilamellar film swells up and large lipid vesicles form that contain all of the cytosolic components that were originally in the bacterium. So here's what phosphatidylcholine looks like. That's the one that I used to make liposomes with here. It's got two fatty acids. One of them is usually unsaturated and puts a little kink in the chain. Those two are attached by ester bonds to glycerol, and the phosphate then of the glycerol phosphate is attached to choline, and that's phosphatidylcholine. Very common lipid in many eukaryotic membranes, and this is the one that we chose to use in this demonstration. So what we do is to add water, just like I showed you, and we end up with self-assembly into lipid bilayers, which take the form of what you see here. This is a computer model of the hydrocarbon chains in the gray in a lipid bilayer. The head groups are on either side, and all the little red and white dots are water molecules. So that's really what a fluid lipid bilayer looks like. That's what we carry around in our heads as a model of a lipid bilayer. And you're going to see a lipid bilayer as a vesicle in the next two slides. Now then, remember that suspension of lipids that I showed you after I added water to the dry film? If I put that under the microscope, this is what you would see. So this is 5 milligrams per milliliter. In this case, this is in 20 millimolar potassium chloride, but I put it into water. And notice that the bar, the space bar at the lower left is 40 micrometers. So these things are pretty big. They range from around 10 micrometers all the way up to about 20 micrometers. And by the way, that is the size of a typical cell, but these are not typical cells. These are multilamellar structures, as you're going to see in the next slide. So here is a freeze fracture image of one multilamellar vesicle. And you can see that it's probably about 10 microns across, and you can see all of the layers of lipid, almost like an onion, that go down right to a small core of encapsulated volume in the center. So that was the original bangosome, the multilamellar vesicles produced by Alec Bangham when he first began to work on this in the mid-60s. So what we learned to do then is to sonicate those in a bath sonicator, and that's what I showed you. You end up with small unilamellar vesicles, and this is what they look like. So after about 30 minutes of sonication, those large multilamellar structures have broken up into these small unilamellar vesicles. These are smaller than bacteria. They're only about 50 nanometers across. See that 100 nanometer bar. Some of them are that large, but most of them are down about the size of a virus particle, 10 to 20 nanometers. Okay, so here's this trick of evaporation. If you evaporate a dry lipid film, or if you dry a mixture of amphiphilic molecules like phosphatidylcholine, along with solute molecules like that DNA, the solutes are captured between layers of that multilamellar matrix. So over to the left, you can see little blue circles. Those are the lipid vesicles. And you see little round things. Those are the solute, whatever it might be. When you dry it down, the lipid vesicles fuse to form a multilamellar structure. Then when you rehydrate, you capture up to half of the original cytosolic components or the DNA in our case, inside the vesicles after they have swollen. So let's take a look at what that looks like. Here is DNA encapsulated after a single wet, dry, wet cycle. The DNA has been stained with acridine orange, very common stain for DNA, and you can see the glowing vesicles in which the DNA has been encapsulated. So all we're going to do toward the end of my talk is to run this same process with the cytosolic components of bacteria. By the way, here's the proof that we actually do capture the compounds, the solutes between lipid bilayers in a lipid multilayer. This is work done with 𝗕𝗂𝗶𝗹𝗶 at McMaster University. We published it in 2013. Reinstadter has a very nice X-ray diffraction setup in which an X-ray beam is bounced through a film on a surface rather than through a crystal in the usual X-ray diffraction method. So this film then, if there's ordered components in it, sort of a crystal array of components, will produce a diffraction pattern, and you see that in the blue off to the right. That can be analyzed by standard X-ray diffraction techniques to give a near atomic and certainly molecular resolution of the material in the organized matrix in this case. So there's the glycerylphosphocholine. This is dimeristoyl rather than what I use. There you see AMP that we're going to trap inside the lipid vesicles. That's adenosine monophosphate. And we put it down through a wet-dry process, just as I did on this slide, okay? Then we shine the beam through it and get the diffraction pattern. And Laura Topazzini did her PhD thesis, and this is her publication. The AMP by X-ray diffraction shows that the AMP has produced a monomolecular and sometimes bimolecular array, a paracrystalline array between the bilayers. And you can actually see down in the lower right the organization of two of the AMP molecules that are right next to each other in this film. And we've shown them here after a condensation reaction in which a ribose on one of them has reacted with a neighboring phosphate on another to produce, in fact, a dinucleotide. And what we have found, I won't have time to tell you about this, but what we have found that in this dry state with a lipid resin, we can get polymers of nucleic acids just by this simple wet, dry technique running multiple cycles. So that's why we did this experiment, in fact. So here we go now. We're going to do this with bacteria. So we start out with live bacteria, in this case, a motile bacterium, as you can see there. We add lysozyme, which dissolves the cell wall and leaves behind what is called a protoplast. Protoplast is pretty fragile. You can either shear it by shear force, or you can use osmotic swelling, and the protoplasts swell and finally lyse to release their cytoplasmic components into the surrounding solution. And of course, two of the components are shown here. The circular DNA will be preserved. If you're careful with this process, you'll have whole DNA molecules coming out. And of course, the ribosomes come out, no problem there. And all that white stuff is a cloud of the thousands of different kinds of enzymes that are part of the metabolism of a typical bacterial cell. So we're going to take that cloud with those components, there it is, and we're going to add lipid vesicles, sonicated vesicles, just as I've shown you here. Along with the little round orange circles are a plasmid containing the GFP gene. And we're going to see if we can reconstitute protein synthesis by going through this process that I just told you about. So we now have a mixture of the lipid vesicles, all the cytosolic components, and we're going to dry it down and notice that we've encapsulated the components within alternating layers in the lipid matrix. When we add water, the lipid multilamellar lipid swells, liposomes are produced, large liposomes, and they contain those cytosolic components. If protein has been synthesized from the GFP plasmid, we should see the resulting cells light up when illuminated with the UV light. So here's the result. This is my second to the last slide. Here is our creature under the microscope. This is a phase contrast microscope of aggregated lipid vesicles after they've gone through this wet, dry, wet cycling. And here they are illuminated with the exciting light showing GFP has been synthesized within those encapsulated components. So what do we conclude from all this? We had to stop there, by the way. This is unfunded research. Rachel Lewis was just working with a little buddy that Lynn Rothschild had available. So we just decided to try this out. So this is up for grabs. Anybody that wants to play around this idea, we welcome you to do that. So we think that we did partially reassemble bacterial cells by this very simple wet, dry, wet cycle. We know that protein was synthesized from the GFP plasmid gene because we can see it glowing within the vesicles. But the question now is, can they also translate some or most of the genes in their genomic DNA into proteins? If so, there's possibility that the systems that make a bacterial cell run in those will be in those reconstituted vesicles and might come closer to fulfilling the promise of build a cell. No one knows for sure this will work, but you are certainly welcome to try. Okay, I'll now get out of sharing my slides, and I hope that we are back and it's time for questions and maybe even some answers.
Thank you very much, Dave. That was really awesome talk. You have some questions in chat. Can you see chat? You have questions in Zoom chat. Can you see that?
Let's see. I do see chat. Is that what you mean?
Yep.
Okay, I will click on chat. Oh, Justine Pearson asked, can those vesicles divide if you encapsulate the bacterial genome? Well, that's the question, isn't it? We don't know how many of the genomic genes would in fact be translated. And I really think I would love to try that. Lynn and I actually attempted to get a Templeton grant to support this research, but we just didn't make it in the year that they used up all their money doing other stuff. So we would have been working farther on this, but where we are right now, you've seen the end result. So we first need to know if other genes are translated in these systems. Then we can ask and perhaps answer Justine's question, whether these could actually grow and divide. From Emmanuel asks, what's the most complex cell you could reassemble like that? Could you do eukaryotic? Well, we chose against eukaryotic just because that is so incredibly complex. We had to work with something simple to start with. And the simplest cell that we had easily at hand was the E. coli and the Dinococcus radiodurans. So we don't know whether we've actually reassembled other systems. For instance, could these cells make ATP? Do they have electron transport systems working? there's some easy questions that could be answered, but boy, it's going to be a year or two of a postdoctoral career to get to those answers. So I think we should stick with bacteria for a while. Do they make new membranes? Well, they won't make new membranes unless the lipid synthesis system is intact. Now, some bacteria will actually take up lipids from the medium as nutrients and turn them into membrane-forming lipids. So if those relatively simple systems are intact, we could feed them in the medium with nutrients of fatty acids, and they might be able to use ATP, which they'd have to make. They'd have to get ATP from someplace to drive this. And if they could make new phospholipids, those would get embedded in the membrane and we could see the membrane grow. And by the way, I've done this with an acyltransferase. I put together an acyltransferase, this is published work, together with lysophosphatidylcholine and oleoyl-CoA, which is the activated form of a fatty acid. And we could watch membranes grow with a completely clear solution. Over an hour's time, we had lots of membranous vesicles growing. Anybody's interested in that, send me an email and I will send you the references. Avery asked if you can use non-lipids like a protein membrane. Well, who knows? If this system can make new lipid and can also make new protein, some of those proteins would have the ability to become embedded in the membrane, or at least attached to the membrane. For instance, maybe these things make some cytochrome C. That'd be easy to test. Cytochrome C is a cytoprotein that binds to the surface of electron transport membranes, such as the cell membrane of bacteria. So somebody should try that. Let's see if that works. It'd be interesting, Elias says, it'd be interesting to supplement the cytosolic components with green lights and analyze the mass spectrometry, whether genomic genes are translated. Well, that'd be a good way to get at that. I think I would pick a relatively simple protein, though, that has its own spectrum, because cytochrome C, very simple protein, 13,000 amino acids, I think, 13,000 molecular weight, I'm sorry. And it's got a color. So we would be able to see cytochrome C very easily by spectrophotometric analysis of the system if it was being made. So I would start with a relatively simple protein. David Nordbjörk asks, can this experiment be used as a model for the original life? Oh, yes. Could you model life starting in different environments? Maybe answer the question whether for the first cell was made in fresh or salty water. Well, boy, those are heavy duty questions. Of course, we are trying to put together a primitive living system, and we're trying to do that starting at a very simple level. Can we make nucleic acids in the system? And we have published papers claiming that we can. People are a little skeptical, and all I can say, try it yourself and see whether you can't make your own nucleic acids. What we do is put in ordinary mononucleotides, just like I showed you in those 2 slides of X-ray diffraction. But instead of just letting them sit there, we add a little bit of heat energy to add activation energy. It turns out that the AMP will undergo condensation reactions in the lipid matrix. Water is lost. The ester bonds that are formed allow polymers up to 100 nucleotides in length to be formed that we can see in gels. So try it yourself if you're interested in this question. Certainly this system is that I just told you about is a model of the origin of life. We're trying to model that with systems like this. Angelica asks, can you use other lipids? Well, yeah, what I just showed you was done with bacterial lipids that we had isolated. We could try this with oleic acid or other simple fatty acids. 𝗕𝗕𝗶𝗶𝗶𝗶 must know that fatty acids are the simplest membrane-forming amphiphile. So we have easily shown, someone should try this. Take some oleic acid, stir it up with some DNA or RNA, whatever you want to capture. Put it through a wet-dry cycle. You will find the RNA or DNA in large vesicles. For sure, this works. So the answer to Angelica's question is yes. It would work with other lipids. Can we select the cytosolic components manually as a medium, sort of sticking in one after another? Yeah, that's what we are doing. You know, we can reconstitute. I'm talking about stuff that other people have done 10, 15 years ago. This was first done by Victor 𝗕𝗂𝗶𝗹𝗱 and Albert 𝗕𝗂𝗶 years ago now. And I was startled by their wonderful paper in PNAS where they put E. coli translation factors into lipid vesicles and showed GFP synthesis. Then 𝗕𝗂𝗕𝗓𝗓 went even further. He got all the way from genes to GFP in some of his systems. So this is being done step by step, but nobody has yet gotten metabolism going in a system like this that could make ATP available to drive all of the necessary energy-dependent reactions of the cell. So that really is a huge hurdle for us to overcome, is to get the metabolic enzymes into a working system in one of these reconstituted cells and get them to the point where they can take in nutrients and do some of the bioenergetics of a cell, electron transport and ATP synthesis. Okay, the last question I see is, no, there's more coming.
Questions keep coming.
Yes, great. From a Chinese word that I can't translate, can we make synthetic vesicles composed of membranes and the cytosolic components that come from different organisms? Well, yeah, there's no reason why not. This would work with any molecule, and I just showed you it works with really big molecules of DNA. This is simple, it does work, try it out, see for yourself. There's a few little technical details. If you email me with any questions, I can send you our original papers in which we did this starting in 1983. We've been doing this all the way back, first papers in 1983. We had a patent on this, in fact, which has lapsed. No companies picked it up. There were other and easier ways to get it into industrial grade liposomes. So it goes way back, and we've had a lot of experience to pass along. Okay, was GFP synthesized even outside the liposomes? Probably yes. We did not try to isolate the liposomes from everything that was still outside, but the GFP that was synthesized inside accumulated in the inside. So we had fluorescence contrast. It was concentrated inside, dilute outside. So it was the insides that had the heavy glow that you could see. How does lipid composition influence the structure of the vesicles? Well, let me take that question from Aaron Agmon to everyone. How does lipid composition influence the structure of vesicles? Very much. We could not do this with long-chain saturated lipids, such as 𝗕𝗓𝗄𝗏-phosphatidylcholine. It's a solid at room temperature. It's frozen. It doesn't work. But if we add one oleic acid to it, so you get what is called palmitoyl oleoyl phosphatidylcholine, it's a lipid membrane that is fluid. It will work because it is a fluid at room temperature. And most people like to work at room temperature, so that's really why we go into that. So lipid composition does count. Some compositions, to go on with Aaron's question, some compositions are more prone to make smaller, larger, or different shaped vesicles. Yes, phosphatidic acid is very highly negatively charged, and it will make smaller vesicles than phosphatidylcholine. Phosphatidylcholine is neutral, has both a negative and a positive charge, and it tends to make large multilamellar vesicles. But things like phosphatidylserine, which has a net negative charge, and phosphatidic acid will make smaller vesicles more easily. Can they demonstrate different permeabilities? They sure can, depending on what you're measuring. We've done a lot of work with permeability, all the way from protons to things like amino acids. And the amino acid permeability is 8 orders of magnitude less permeable than a typical permeable species like water. So that is the range of permeabilities across lipid bilayer membranes. So you really have to keep that in mind when you're thinking about permeabilities of membranes. And it's a whole study all by itself. We have one book out on this called Membrane Permeability, I think, and it was published, a multi-author book, edited about 10 years ago. Okay, any other questions here? Yeah, I see one more.
One more, yeah.
Okay, are we finished then, Kate? Do we have enough time?
Yeah, I think we still have 10 minutes on schedule, so you can answer the last question easily.
If there are hydrophobic peptides during a liposome dry up, do they get included in the lipid bilayer of the rehydrated liposomes? Or perhaps they show some function on the membrane? Yeah, we did this years ago. In 1994, we had a paper in Biophysical Journal in which we dried ordinary phospholipids with polyalanine or polyleucine. And in certain conditions, rather rigorous conditions, we were able to get those completely nonpolar peptides into the lipid bilayer. And they did impart a function to the bilayer. Because those chains were in the bilayer, they made a hydrated defect through which ions could flow. So in fact, protons were more permeable than ions like potassium or sodium. We actually measured those. So it's really interesting, we thought, and nobody else has picked up on that protons can be made permeable to membranes by putting these nonpolar peptides into the lipid membranes. That was published in 1994. The first author was Ann Oliver, and this is her PhD thesis in my lab at the time. Somebody says, thank you, and I say you're welcome. Possible to perform reconstitution with organelles like mitochondria. I've tried putting chloroplasts, not mitochondria, but we are trying quite recently to get working chloroplasts into these cells so that the chloroplasts are actually surviving and can function in the cell. Now, the neat thing about that is that chloroplasts can use light energy to make ATP. So we might put together a kind of a little plant cell using photochemistry and ATP synthesis to provide ATP as an energy source for all the metabolism. So we've given that some thought, and I would like to think that a chloroplast could be encapsulated because it's such a gentle procedure. Just dry it down, rehydrate. Things, life does this all the time. What do you think a seed is? A seed is a whole bunch of dry cells. And when you wet it up, it all comes back to the life. Kate, do you have any questions?
I don't. I think you answered most questions that I would have. And I just want to say you make it sound so easy. Like when you talk about those experiments, you make it sound like it's really easy to do. But I know from my own experience that it all requires a lot of finesse, a lot of experimental skills. So yeah, just for people that haven't done it.
I did mention there's some arts and craft to this, and I'd be very happy to, clear up that kind of thing. If people send me an email, you're welcome to.
thank you very much. Thank you so much for giving a talk, and thank you for answering all the questions.
Good. It's my pleasure. Thank you all for coming.
And if anyone wants to ask Dave any more questions, you can email him. So thank you very much, and see you all next week.
Okay, bye now.
Thank you. Bye.